Okay, so before I was so rudely interrupted by iPhone storage reminders and had to go through and that whole process, let's get back to what I was talking about before I was cut off. So, specifics in terms of what you need to know or who you need to know enzyme-wise in DNA replication. You don't specifically have to know DNA primase and RNA primers, but they are integral to the process because they provide a starting point for where DNA polymerase is going to begin the journey of adding the complementary base pairs. DNA primase is going to attach, add these little primers, and that's going to allow a starting point for DNA polymerase to know where to begin, because during this process, there's going to be many replication forks that open up in the DNA molecule in order for it to get copied or replicated in a quick manner. Without that happening, if it just was one replication fork, you can imagine how long it would take for those 6 billion base pairs to be replicated or read by DNA polymerase. So it's going to have many replication forks for this process to begin. So let's take a look at DNA polymerase. So DNA polymerase are adding the complementary base pairs, adding a T to an A, adding a C to a G. And if you look at this 5' here and this 3', in terms of the leading strand, the replication is going to follow helicase. Helicase is going to go toward the replication fork. The leading strand is going to replicate with the replication fork from the 5' end to the 3'. You're adding the new bases. If you look at the lagging strand is going the opposite direction because remember, the strands run anti-parallel, and it's going from its 5' to 3'. Okay? So we're going toward, I think I did this one opposite. But here is our 5' we're looking for leading. I circled the wrong one, I apologize. Here's our 3'. So we're going toward the 5 here. We're going toward this 5 here. So those are your 5s you're looking at. Here's one, we're going toward this 5, we're going toward this 5. You watched in your Edpuzzle video that there are some segments that are added to the lagging strand in chunks, and those chunks are called Okazaki fragments. They were discovered by a Japanese scientist who named them after himself because that's what gets to happen in science, but he was able to recognize the replication process and see that they were added in these small segments in order not to overload DNA polymerase going backwards. So the leading strand goes toward the replication fork without stopping. The lagging strand is going backwards. You also know about the final player in this whole case, which is DNA ligase. Ligase is going to follow behind polymerase. You can see it here. It doesn't show it on this strand, but what it is doing, it is initiating that hydrogen bonding between the two strands. So it's going to help this from degrading or coming apart, or all the hard work of DNA polymerase. Okay, so that's going to help initiate that bonding between those A's and T's and C's and G's. So one thing that DNA polymerase is doing that's not only adding the complementary base pairs, it's going to proofread the strands. Okay, so once the base pairs are added, and mutations, which we'll get into soon, are not a big component of DNA replication. It happens a lot less than you might imagine, but DNA polymerase is also going to proofread those strands to make sure that there are no errors before it heads out of that S1 checkpoint. We learned about those checkpoints. We wanted to make sure that everything was ready to go before we go into the mitotic divisions, and this is an integral part of that checkpoint, DNA polymerase proofreading the strands.